Hello everyone, I'm Anas Al Ubaidi, and this is lecture 16 or session 16 within chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. The chapter is about conceptual and theoretical monitoring, while the topic for this session is about monitoring of a drug idea and drug conceptualization interaction trials. Introduction. Definition. As drug idea and drug conceptualization interaction trials analyze how hypothetical the drug designs and theoretical treatment principles are translated into actionable, testable clinical realities. These trials focus on the interaction between conceptual frameworks and the clinical application of new drug ideas. Key points. Patient safety. Why is it critical? When moving from a theoretical concept to practical application, unforeseen safety issues often arise. These trials must anticipate and address potential risks prior to widespread testing begins. Here's a summarized diagram on ensuring patient safety in drug trials. As here we have pre-screening challenges, safety monitoring, and off-target effects from the left to the right. Key actions for ensuring safety. Comprehensive pre-screening early trials are conducted on carefully selected populations. To minimize unexpected adverse effects, pre-screening identifies participants who may be more susceptible to theoretical risks, as for example, a conceptual cancer drug targeting angiogenesis was pre-screened in healthy volunteers to assess the vascular side effects prior to introducing patients with malignancies. Proactive safety monitoring frameworks. Each exploratory study must establish safety oversight committees with real time reporting capabilities to manage adverse reactions. As, for example, an early phase CNS central nervous system drug trial identifies mild inflammatory responses in the brain leading to immediate therapeutic adjustments. Detailed documentation of off target effects, unexpected drug impacts, such as those on unrelated organ systems, are captured for refinement and redesign if necessary. Data integrity. Why it matters. The success of a drug conceptualization relies on reliable, high quality data to confirm or challenge theoretical predictions. Mismanaged data can skew results, causing delays or failure. Here is a summarized diagram on ensuring data integrity in drug trials. On the left, drug data integrity, and on the right, standardized collection methods, regular data audits, use of validated metrics. Strategies for ensuring data integrity. Standardized collection methods employ uniform procedures to gather and process data across multiple trial sites or centers, ensuring comparability. For example, a neuroprotective compound was judged based on standardized cognitive assessment scales like MoCA to validate its effect consistently. Regular data audits, regularly reviewing and verifying trial data eliminates inaccuracies or inconsistencies that could compromise the trial's outcomes. As for example, early testing of a novel immunomodulator used site-specific data reviews to ensure biomarker variability did not result from measurement error or bias. Use of validated metrics, novel endpoints like inflammation biomarkers for anti-inflammatory medications are measured using validated tools to ensure reproducibility. Protocol adherence. Importance for trial outcomes. Strict adherence to protocols ensures that drugs are tested under conditions mirroring their conceptualized design. Ensuring adherence. Clear protocol design. Protocols are designed to mirror the theoretical models, specifying all parameters like dosage intervals, route of administration, and patient monitoring. Example, a conceptual long-release chemotherapy protocol ensured administration intervals aligned with pharmaceutical release kinetics. Regular reviews. Periodic protocol reviews allow for minor adjustments without deviating from the trial's primary objectives. And here is a summarized diagram on ensuring protocol adherence in trials. From left to right, we have clear protocol design and regular reviews. Regulatory compliance. The rule of regulation, regulatory agencies mandate robust conceptual trials to safeguard patients during the transition to clinical phases. Non-compliance can halt all further development. Essential regulatory practices, filing comprehensive investigational applications, submissions such as IND, investigational new product, drug or product applications, require detailed justifications, risk analysis, and thorough trial designs, as for example, a metabolic drug targeting dual pathways provided preclinical animal model data to gain first in human clearance from FDA. Engaging with regulators, continuous communication ensures alignment with rapidly evolving guidelines, critical when testing groundbreaking drugs or medications. And here is a summarized diagram summarizing or showing navigating regulatory compliance in drug development. And here on the left, we have regulatory compliance. On the right, towards the right, the role of regulation, essential regulatory practices, and engaging with regulators. Steps to effective monitoring. Step one: site selection. Prioritize academic or research sites with advanced technical capacity and expertise in early phase trials. Example: translational centers specializing in phase 1 biologics hosted trials for an immunotherapy drug with highly specialized endpoints. Step 2: risk-based monitoring (RBM). Devote additional monitoring resources to areas with high complexity or risk, as for example, a conceptual Alzheimer's therapy trial focused data collection on participants with rare genetic inflammatory predispositions. Data review. So step 1 was site selection. Step 2, risk-based monitoring (RBM). Step 3, data review. Analyze real-time deviations from theoretical models and update designs accordingly, as for example, an extended release analgesic trial modified its plasma release predictions based on early human PK/PD data, pharmacokinetic/pharmacodynamic data. Step 4, reporting clearly delineate conceptual predictions versus real-world findings in research documents for regulatory bodies, enabling informed guidance for ongoing trials. Tools and techniques for monitoring. Electronic data capture (EDC) systems centralize trial results from multiple locations to ensure quick access and cross-comparison. Example, an EDC system integrated plasma concentration curves with self-reported side effects in an antihypertensive trial. We have simulation and modeling tools. Use computational models like PBPK, physiologically based pharmacokinetic modeling, to forecast organ interactions. As for example, PBPK predicted kidney toxicity in a conceptual renal drug, leading to preemptive reformulations. Biomarker and endpoint analysis validate novel biomarkers developed during conceptual stages, refining their real-world relevance. Example, a neuroprotectant study tracked cellular pathophysiology biomarkers for brain region specificity. Interim statistical analysis deploy early phase statistical modeling, as for example, Monte Carlo simulations to identify variability trends prior to larger scale trials. As an example, interim statistical reviews refined the pharmacological dosing range for a sustained antidepressant release schedule. Practice applications and lessons learned. Examples of successfully monitored trials: metabolic dual pathway drug, preclinical and phase 1 trials validated simultaneous safety profiles for hepatic and cardiac biomarkers. First-in-class vaccine for rare viruses, early trial stages aligned almost perfectly with conceptual predictions of its immune suppression capabilities. Extended therapy for CNS central nervous system disorders, data consistently validated theoretical drug release timing, reducing trial design costs later. And here is a diagram summarizing the successful monitoring in drug trials, as on the left, successfully monitored trials, on the right, metabolic dual pathway drug, first-in-class vaccine for rare viruses, extended therapy for CNS disorders, central nervous system disorders. Next, we will summarize what we have covered through this lecture through the following via the following tables. Um, Table on key differences between drug idea and drug conceptualization interaction trials. Aspect for each aspect, the comparison between drug idea and drug conceptualization interaction. In the aspects, we have the definition, trial stage, stakeholders, example, application, goal. Table on key components of monitoring, component focus example, components we have patient safety, data integrity, protocol adherence, regulatory compliance, concept validation. Table on steps in effective monitoring, step purpose example, steps we have site selection, risk-based monitoring, RBM, data review, implementation, real-time adjustments. Table on tools and techniques for monitoring, tool or technique purpose example, tools technique we have EDC, simulation models, biomarker validation, term statistical analysis, visualization software. Table on real-world applications of monitoring, trial, conceptual focus, outcome trials. Here we have a metabolic drug study, anti-inflammatory therapy, vaccine development, pain management therapy, and CNS central nervous system neuroprotectant. Application table, scenario for each scenario, care principle applied, key actions, timeline, what to watch out for. As scenarios, we have transitioning a dual-acting antihypertensive from theoretical model to clinical prototype, validating safety of experimental cancer drug targeting angiogenesis, exploring neuroprotectants for CNS central nervous system diseases within a conceptual study, ensuring dosage precision for extended release anticoagulant designs, studying dual pathway metabolic drug safety and efficacy in stratified groups, comparing drug clearance rates with PBPK simulations during human trials, testing anti-aging drug biomarker during longitudinal trials, implementing combination therapies for inflammatory disorders targeting cytokine suppression, evaluating synthetic vaccines with integration of immunogenicity markers, cognitive validation studies designed around CNS, central nervous system drug designs, tracking real-time absorption with sustained release antidepressants, flagging adverse trends in conceptually driven multi-mechanism therapies. Next, we will revise what we have covered through this lecture through some multiple choice questions or MCQs. Question 1. What is the primary goal of drug ID and drug conceptualization interaction trials? 1A. To generate new molecular compounds. B. To test off-label uses of existing drugs. C. To bridge scientific hypotheses with clinical applications through actionable trials. Or D. To conduct large-scale studies on drug affordability. The most accurate correct answer in between the four choices is C, to bridge scientific hypotheses with clinical applications through actionable trials, as these trials aim to transition theoretical drug ideas and treatment frameworks into practical, testable clinical protocols, ensuring safety and efficacy through systematic evaluation. Question 2. Which monitoring component ensures drug prototype designs are executed as envisioned? A. Patient safety. B. Protocol adherence. C. Regulatory compliance. Or D. Data integrity. It is B as a protocol adherence. So B would be monitoring component to ensure drug prototype designs are executed as envisioned. It would be C. It would be B. Protocol adherence, the adherence to the protocol. As protocol adherence ensures that clinical trials follow predefined conceptual designs and methodologies without deviation. maintaining the validity of the theoretical model being tested. Question 3. What tool is crucial for refining conceptual designs during early trial phases? A. Paper-based records. B. In-term statistical analysis. C. Handheld sensory probes. Or D. Adverse event logs. So it is B, as in-term means ongoing trials statistical analysis. So the statistical analysis for ongoing yet still trials, not ended yet. So B would be the answer. In-term and statistical analysis enables researchers to analyze data trends and adjust theoretical models, ensuring alignment with real-world data while maintaining the integrity of early phase trials. Question 4. Why is regulatory compliance critical in drug ID and drug conceptualization trials? Uh, A. To secure approved protocols for innovative therapies and ensure patient safety. B. To eliminate the need for endpoint monitoring. C. To avoid focusing on biomarker validation. Or D. To bypass preclinical testing requirements. So the most accurate answer would be A. To secure approved protocols for innovative therapies and ensure patient safety or participant safety. So A, as regulatory compliance ensures that trial designs adhere to safety and clinical standards such as IND, investigational new drug applications, while enabling transparent alignment with FDA in USA and EMA in European Union in the guidelines. Question 5. Which example demonstrates the effective use of simulation tools in conceptualization trials? A. Manual blood pressure recording during drug trials. B. PBPK models predicting organ-specific drug impacts before in vivo testing. C. Verbal feedback from patients on dosing schedules. Or D. Separate sheet-based pharmacokinetic analysis. So it is B. The correct answer as B. PBPK models predicting organ-specific drug impacts before in vivo testing. As physiologically based pharmacokinetics or pharmacokinetic models simulate drug interactions across organ systems, offering significant insights into theoretical impacts before clinical trials begin, reducing uncertainties and optimizing designs. And here we come to end of this lecture. Thank you very much and goodbye.